All right, good morning, everybody. Thank you. Good morning, everybody. Today I'm going to discuss the biomedical applications of octopuses, specifically in how they pertain to engineering. Briefly, I'm going to go over their anatomy, any relevant background information, along with, and then briefly go through, summarize 3 papers that pertain to this animal. And hopefully, we could I can elucidate some of the relevance of this animal model for some of the research maybe we're doing here. All right, first of all, going over the anatomy itself. So this the photo in the left photo here is actually the main central brain. So typically they have 9 brains. They have one so it's kind of like the main brain that's located in the mantle of the octopus, which is the balloon sac at the back. And then they have 8 ganglia that house at the base of each tentacle itself. These ganglia do regenerate, as I'll discuss later, but and essentially they do operate independently, but also with feedback to the central brain here in the photo. So as you can tell, I also want to point out that this evolved independently without any need for a spinal column, because this is an invertebrate animal, which is interesting too.
What is the right picture?
The one on the left.
What is the right one?
The right. So I'll get to that third.
Okay.
But yeah, it's cool. It's an interesting thing too. So these with each glia with each ganglia on each one of their arms, they have their suction cups actually have olfactory sensations, and they have chemosensors, and as I'll go over briefly, photoreceptors too on their tentacles themselves. So the information that is received from each tentacle is fed into each ganglia in the arm. That then is processed and almost supposedly maybe an executive function way in the central brain itself, which is I think is pretty cool. As discussed previously, more convergent evolution that I think is really interesting and can probably be shown more for the anatomy is the octopus does have a beak because it has a radula, as it is a mollusk. So the radula here is in the right photo. If you ever seen like a snail, like have one of those, a snail at an aquarium, and you see like kind of like scrape the algae off the side of your aquarium, that's the radula. It's like a cat tongue almost. Because it's kind of more like sandpaper. They use it for octopuses themselves use it for opening crustaceans or anything hard or other mollusks like clams. And it also is also worth noting that their saliva is actually toxic too. It's kind of like at the spider level, so it's like so the therapeutic index is low for most octopuses. There's certain ones that are deadly to humans, but I mean, otherwise it's like maybe part of the spider. It's not that much. It's not that venomous. So, additional to the anatomy that I think is relevant for this presentation, they have 3 hearts. They have one by each of their gills. That's located by the organ they use to store water as well. So these octopuses can store water to briefly go outside of the water, and almost like a scuba way, where they kind of hold water close to their lungs. And because of this, they have additional hearts for each gill that then helps pump that water through their gills. And then they have like a main heart in the center that's again located underneath in their mantle, underneath the brain. And this helps pump blood to the gills, to the hearts that pump the blood from the gills. And another relevant anatomical feature that I'm going to discuss too that's relevant for the studies that we're going over is the octopuses do possess chromatophores on their skin. These chromatophores are cells that are not really cells, but they're a collection of cells, you could say, because there's glia associated, but I'll go on that more on that later, that help the octopus change color with different pigments. And of course, what's relevant to neuroimaging itself is instead of hemoglobin, or globin, I should say, that humans possess, they have hemocyan, which is actually, which I'll go over briefly, I think, in the next slide, but it's also really important for some of the imaging papers and some of the critiques I had on that. So briefly, before I go into hemocyan, I'm going to go over the chromatophore. So
Can I ask you a question?
yeah, sir.
So hemoglobin, I think, is because the last part is for iron, right?
Yeah, so heme is the functional group that's in the middle of these two well, four, there's two different subunits that then attach and kind of form this ring. There's like an alpha subunit and a beta subunit, and they and it's you know, there's four altogether because they cluster, right? But then like there's alpha, beta, and in the middle is that theme group, and then a theme group is where the iron kind of is housed. And it's an indirect way of transporting oxygen to human beings. I'll definitely go over that later though. And in much more detail, and it'll also pertain to why in an imaging standpoint, it's kind of a little different for a different molecule. But yeah, long story, yeah. So chromatophores themselves were first identified and described in a 1970 paper. I know it seems really relevant that we just didn't even understand why these octopuses were changing color. It's a neuromuscular ligand itself, so it does use action potential, but it also has ligand activated channels too. So it's a really complex organelle, I guess I would call it. It produces different pigments. These pigments themselves are tryptophan macromolecules themselves that oxidize in a way that produce a different color. These colors themselves are actually differ among the species of octopus. So the common octopus, Octopus vulgaris, has orange and yellow mostly, but chromatophores or the pigments oxidize in an orange and yellow fashion. However, if you go into my favorite kind, the giant Pacific octopus, they oxidize in more a navy blue area, which is interesting too. Squid oxidize more pink and red. And it's hypothesized that why this is important is it's hypothesized that these chromatophores are not only used for evasion, but also perhaps language and communication as well. There's been a recent study that showed that I think I can't remember the type of octopus, but where they actually had social groups themselves, the most social octopus, and it was ignored for like a couple of decades until recently, where we looked into this, and it's going to be pretty huge that they seem to communicate mostly like the squid with the chromatophores, which is interesting because these chromatophores, as I previously described, are hooked up to have action potentials and ligand activated channels. So there's obviously a lot of brain effort to actually an evolutionary investment in these chromatophores themselves. How these pigments vary and differ depends, I mean, as I described, based on the species. And also this cluster density differs depending on the animal as well. And that's how they can control the color spectrum from like really red, really pink to like navy and purple, if that makes sense as well. So as we can see from our eye, they're like in the same way that like our computer screen works, where it's a whole bunch of red, green, and blue from the computer screen, like at a tiny scale. From a much larger scale, our brain kind of wants to simplify it, and we use heuristics and shortcuts to make it look like it's a different color than it actually is. So it's a similar mechanism that the octopus uses based on these varying pigments and these subtle changes. Now going into the hemocyan molecule. So there's actually different types of, there's like, I think it's like roughly 5 different types of blood we can see from an evolutionary standpoint. Most vertebrates themselves develop hemoglobin, and it's an indirect way, as I previously described, of exchanging oxygen through the lungs. There's a Van der Waals effect. So essentially what happens is we have inspiration, where we inhale air into the trachea, into the lungs. The alveoli essentially have a Van der Waals bond that then transitions the oxygen to the hemoglobin molecule itself. And it's not directly connecting, it's indirectly connecting, but through that exchange is a more efficient at our room temperature. However, if you look from an evolutionary standpoint, if we're underwater, it's a lot more cold, and the pH is a little different. Then the efficiency of hemoglobin actually is not the best. Actually, hemocyan is actually a lot more efficient, and how hemocyan works is it's a direct bond that actually occurs with oxygen, It has, I mean, depending on the animal itself, arthropods have a lot more subunits, but mollusks themselves have like a more circular subunit, which then directly bonds to the oxygen themselves through the gills in their heart. And what's interesting about, and what's crucial to say about this from an imaging perspective, is that this metal bonding is actually not done through heme or well, it is some a type of heme, but not iron specifically, but copper. And what's interesting about this copper is it has like iron, it has a really interesting D block orbital. Not to get too much into quantum mechanics, but because of this orbital itself, it makes it an interesting choice to bond oxygen molecules at such a low temperature. If you look at the structure of this molecule itself, it's circular. And the subunit, it actually has a dipole interaction because it does need to work with a highly electronegative chemical like oxygen itself. And one of the reasons that we hypothesized that people ran experiments that found the efficiency of oxygen transfer and exchange rate. And they found that at the sea level temperatures that were the common octopus we found, it actually is more efficient to use hemocyan as opposed to hemoglobin for oxygen transfer. So it's hypothesized that the temperature and underwater conditions allowed for this to happen. But it also, from a imaging standpoint, kind of shows the need for a pH and temperature regulated environment if we're going to study animals or non-hemodynamic or different hemodynamic imaging, any parameters for imaging itself. So there are, I'm going to briefly mention that there are other types of blood. So this hemocyan molecule, because it has copper, looks blue when it is oxygenated and oxidized, which is pretty interesting, as opposed to hemoglobin itself, which is red.
This is the original blue-blooded.
Yeah, it's like a greenish blue, but that's because of the copper. There's other types of there's other colors of blood if anybody else is wondering.
What about fish? What is it? Because of the same temperature.
Vertebrate fish do it as well. They still use hemoglobin, but they have a different mechanism for it. So it's interesting. So this is hemocyans are mostly arthropods and mollusks, and part of it may be just because of convergent evolution. Evolution itself is never perfect. For example, like the same fish have a valgus nerve that goes to vagus nerve that goes from their gills and their brain, and we still have that nerve, but because we don't have gills, we have lungs. It kind of curves around our heart and goes back up to the brain. So actually, fun fact: heart surgeons have a hard time. Well, it's really important that you don't nick that nerve because it deals with inspiration itself. It would be a hard time breathing if you nick that, but it's a very inefficient nerve that we. It would be nice if we didn't have it because it kind of just goes from here, loops around the heart twice, and then goes right back up to the brain. So it just doesn't make really much sense, but it's because of the path of evolution, if that makes sense. So fish had a different path because of the vertebrate. That answers your question.
Does that nerve go anywhere near the guts?
Which one?
Sorry. Does that go anywhere near the guts?
Vagus nerve?
Vagus nerve?
I think so.
No. But yeah, it's you just don't want to nick it, but it's.
No, but my point is like your point was about efficiency of cyanins at the water temperature. Well, fish were there, I mean, as well, it's like evolved there,
Yeah.
so why hemoglobin was discovered, quote unquote, right?
Yeah, evolution's random, right?
Yeah, it's random, but I guess we'll find out, right? It is worth noting that invertebrates were really dominant for, I think it was, don't quote me on this, but the first extinction level event. Because there was nautiloids and what is it?
Got a nautiloid shirt today. It was
Yeah.
accident.
Oh, yeah. You see,
Yeah.
enema, what is it? And then my favorite is the giant shrimp thing, the enema collis or whatever. I'm
Nautilus is the only one that survived, right? The
Yeah.
extinction.
Yeah, most of them are gone.
They got like smaller though. They were a lot larger, but like around that time period, there was a lot more invertebrates. So there would have been a lot and crustaceans. So there would have been a lot more hemoscience. But then, so I guess to answer your question indirectly would be perhaps the extinction level event that happened during that favored hemoglobin in the water briefly. And then, but not to the extent that it would wipe out hemoscience. So,
That's true, yeah, warming event or something, that makes some sense, yeah, it could have happened, yeah.
well, I do think that there was, cause if I remember correctly, it was a solar flare at the time, so that's probably part of it. So there's probably a lot of compounders there that are indirectly associated, so who knows, maybe we're still recovering from that event.
Well, ecology's complicated.
Yeah.
Yeah. But then, but the other colors of blood, just for, cause I thought this was interesting, was, so flatworms and leeches have green blood, and it's, yeah, it's green, it's like gross. And, yellow blood is, well, violet, I have the violet one here, but that one is from, it uses, an iron collection, method when there's a lot of heavy metals, because these worms themselves live right next to the volcanic, kind of, subduction zones in the Atlantic. And so there's a lot of, toxic metals that come out, so your blood needs to kind of filter those through. And their blood's violet, which I think is interesting, but it's because of that color and the collection of metals that need to kind of make sure that they don't absorb mercury or whatnot. Anyway, I think that, so there's obviously a hemodynamic issue with the blood in octopuses themselves. So going over briefly the complication of the complicated tentacle itself, there's typically inside each suction 4 different type of sensors, which all provide information to the ganglia itself for each suction. This is like this figure here is the inside of a suction and one wall of one of the suction cups. So if you can tell, there's most of these are chemotactile sensors that it's not the biggest, I thought that'd be a lot larger in the video. But if you can't tell, they're shaped differently because they are made for collecting different chemical shapes. But the one that is specifically I wanted to point out is a photoreceptor that that's actually looks similar to a plant sensor in the chloroplast itself. But essentially what this does is it bounces off a couple electrons when it has a photo, when it sees light, and then sends a signal down to the ganglia, if you can keep saying glia, the ganglia, through the pathway illustrated. So, and the type 3 photosensor does see fine details of light itself to differentiate on top of the chemotactile senses that are being inputted from the tentacle itself. These tentacles usually run on GTK ligands, very similar to conceptually similar to our ATP cascade that we have in our mitochondria and our cells. So it's a very similar, if you're familiar with that, it's a very similar mechanism where it's like one ligand goes into the sensor itself, it changes shape, it spits out another cascade, and that provides information, and that information then is then collected in the ganglia. Each ganglia itself has its own kind of a node, let's say, for the amount of information that is inputted from whether it be light, whether it be the chemicals themselves. And and we zoom out from the suction cups themselves, we see that each tentacle actually has a whole bunch of like a mesh almost of longitudinal and transverse muscles fibers. And these fibers obviously are really interested from the standpoint of robotics, because there's a lot of properties that like our muscles are not like grouped together like that. They're more attached to our bones and skeletal structure. And the octopus itself has evolved to not have a skeleton really. So instead, it's just kind of like a interwoven spaghetti of muscle, which has its properties, but then also because there's an action potential for each strand that is needed for the muscle, it creates a really complex relationship, but that will go more. I'll provide more depth when I go over the robotics paper. It's also worth noting that these neurons in the tentacles themselves are fully capable of regeneration. A paper I'll go over in a bit shows the extent of how much the regeneration occurs. But as long as the central brain as a whole stays intact, the octopus is still able to regenerate, which is fascinating. But also comparative to our body, we can't regenerate, well, we can regenerate peripheral nerves, but not these muscles. Our muscles do not generate our it's muscle cells and neurons that are that we're kind of stuck with, and I think neurons, there's debate whether the early cells and stem cells actually produce more neurons, so, or can actually become neurons and help with that, but I mean, for the most part, we say that neurons and muscle cells do not you're kind of stuck with those and they deplete. So first, a question for the audience for more engagement. What convergent evolutionary features do octopus share with any other animals? Anybody have any like ideas that were like, oh, this looks like this animal or anything? It's a question I have for the group.
You mean the beak?
Yeah, I mean, that's, yeah, like birds, obviously, right?
Well,
Yeah, beak, yeah.
Beak.
How closely related are octopi and squid?
They're both cephalopods, but I had it on my phone, the kingdom itself. I think it's because cephalopod itself is in the kingdom, but if I go to the hierarchy, it's like cephalopod, octopus, squid, nautiloids, if that makes sense.
Right,
And then there's the vampire squids over here,
okay,
So it's at that level that they diverge.
so,
So the common ancestor would be a little different, I think, at that point.
yeah, so that's not really, so that, the arms aren't really a convergent evolution, like, the whole family has that.
I don't think so, yeah, because every cephalopod has arms. Although nautiloids are really weird with their arms, because they don't have any suction cups, they actually are just like they're a whole bunch of strands, it's not just like 8, there's a whole bunch of them. But like they're short too, but they're like up in that the shell, which is weird, but. So those ones are really weird, maybe that one's the most different, I should say, out of all. If I compared the squid, which have what, 2 main, and I can go over squid brains too, because those are even weirder, because they're donut shaped and they form around the beak. Anyway, I can get too much into that. Any other conversion or evolutionary features?
I guess chameleons also have chromatophores or side, don't they?
Yeah. So yeah, that's a good point.
What about geckos with their suction cups?
They have they actually, so they use different types of suction. They have a van der Waals effect that with water, actually, I think it is. I want to say it's a van der Waals bond, which is, I think, that doesn't make sense though. But I'll have to get back to you on that, I think. I do think that my gut tells me it's van der Waals, because I know that there's one type of lizard that uses the van der Waals bond to run on water. But it's a different mechanism, I think, because it's not really, I don't think it's really suction. Well, it's a bond, but it's not like suction to the extent of an octopus suction, because like octopuses have like all these sensors in their suction cups as well that function very similar to like an olfactory, maybe like a human tongue, in an octopus suction cup. Maybe that's except for our tongue doesn't really like, I don't think there's photoreceptors on our tongue though. So that'd be cool though. But yeah.
I
Okay.
have a question,
Yeah.
if part of the brain of the octopus is on its arm.
Yeah.
And if it cuts, if you cut that, it will regrow, do you think like their behavior will change slightly?
There's been debate about that. There's one paper I didn't include in this one, but there's one paper that tested whether octopuses prefer a tentacle, and it turns out that the octopuses are like right-handed, left-handed like we are, so they do have preference to a tentacle. So I think we don't really know is my answer, but I mean, based on that reference, I would say maybe, because I mean, if I was right-handed and I lost my right arm, I think it would have to be a little bit of a learning curve to try to relearn my left arm. But then if my right arm grew back, I don't know if I would still prefer that right arm.
They did kind of have hemispheres in that central brain, not like in the way that we do, the Clemsen hemispheres, but they had the symmetry there, right? It looked like.
It looks like a hindbrain, doesn't it? That's what my thought was too, except for like, but it's weird because like we wouldn't have like the first animal with the brain, well, it's like brains didn't really, they kind of showed up in jellyfish and ecaphoids, that's when we first had neurons really, because sponges were animals and they didn't have a brain. Depending on where you want to draw the line of what species are, but I mean, yeah, this brain is completely, that doesn't have a spinal column or anything, but then at the same time, it looks like a hindbrain to me, which is like if I pulled up like a shark brain and an octopus brain, they look similar to me, so I don't know.
There's only so many ways to brain.
You can like teach octopus to do something, right?
Yeah.
Small task. I mean, if you cut some of his arms, does he forget what we learned?
I don't think so, because it's in a similar context. It's like if we lose a couple of neurons in our brain, we still have a network that still can kind of back it up, I guess I would say. So it's kind of like if I would have a lesion in my brain, would I still have the capability? It's not like I lose the information. It might be a little slower because the functionality in the network changes, but there's still other neurons that can still kind of form a network. So I think it's a similar context to
It
that.
sounds like suggesting the ganglia are specialized though, is I think what you were more getting at, right?
Oh, is there specialized? There's some debate about that. It looks like more like it's more like a network. It doesn't seem like it's as specialized.
Right.
Yeah.
Like I had like a seeing arm and like a
Yeah,
my sign
that would be-
language arm, you know.
Actually, that will be explored in the robotics paper, because that's my main critique with their paper, is they said, oh, we mimic it. Anyway, I'll get into it a bit, but that'll be answered at that point, because I that's my critique with the robotics paper. So hold on to that, because I think it's a really good question, but now I'm gonna transition, I'm gonna over briefly the autonomy and relevant information for this slide. I'm gonna briefly, let me check the time, I should do about that. And myself, boy. I'm gonna go now into I'm gonna transition into octopuses and biomedical science themselves. So the first stage, and I knew this would be my most detailed presentation. Octopuses themselves were the first animal models themselves in biomedical science was in 16th century. The father of medicine, Alcmaeon of Croton, was the first person who, he's supposedly thought about dissection. He's the first person learned about dissection, used animal models, and recorded the significance of using animals for medical structures. Some of them, briefly, the paper, I guess my presentation will go over these areas of medicine, but there's other areas as well. I will go over robotics briefly, neuroscience, regenerative medicine. I also want to point out that according to my Google Scholar search, did show that the octopuses in biomedical science have been increasing. I think I previously went over that 1970 was the first time that we actually described the chromatophore in the octopus and looked in cataloged its information and had a concept of how it worked. Since then, the interest is, since the past 2 decades, it's been exponential, and the rates of papers using octopuses as models. I will go over briefly some policy implications, I think a little bit later about that, but some of these papers, most of the papers I'll be going over were 2010, in that kind of time frame, 2010 to 2020. But there are more papers in 2020. They just are so new that I didn't know if I should include them. There was one that I think was about AI, and one that is the call for more tanks, so these octopuses can actually live on land, because as I previously went over, their oxygenation is not really the best for land, and they're not really the most social animals, so getting a whole bunch of them together for a good sample size is not really doable currently. And that paper was published 2023, and the AI one was published 2022, but I just didn't think that was I wanted to wait and see what people said about it first before I presented on that. Oops. So briefly going over some robotic implications. In the field of prosthetics, there's an immunological issue as well as a mechanical one. When you lose an arm, they typically have to surgically fold the neurons back in to the nerves back into your arm, and that creates a feedback loop, so you don't have a phantom limb or any other a myriad of neurological issues. There's also then an immunological issue as well, because there can be anything from infection to as well as just an immune response to the metals themselves, since a lot of these prosthetics do use metals and silicon, which is actually, in fact, silicon is actually poisonous for the human body. So there is an immunological component that needs to be recognized in prosthetics. So this paper, so specifically with octopuses, there's been a really big talk about just the biomechanics of the octopus arm itself. Robotics the field of robotics have been having an issue with hydrostatic robots, really, which is really just that interwoven spaghettiness of the octopus tentacle. And so this paper will previously, in the field, they've looked at the reaching movement itself and the biomechanics of the octopus. The formulas themselves here below are one of many that they use to calculate the muscle stress of the octopus itself. This paper specifically, they looked at in the same way that Archimedes dropped a crown in the water and looked at the buoyancy and the water displacement and more that this paper actually did as well. They also looked at the velocity of the water and the drag, and they used those variables to eventually find the formula here. Which if you look at L naught is the muscle length, which is what they use to find when they drop the tentacle in the water. They wanted to see the muscle fibers themselves and the density. And more or less what this equation is doing is it is trying to find the limits of the muscle itself and the of two forces. One that's acting upon within the muscle and one that's acting upon the muscle. And it hopes to find the limit of the amount of force that this octopus muscle can have. Essentially, I include the other formula too, where they or how they got to how they derived, I think, the certain values that they can use for the stresses that they for the octopus muscle itself. This reaching movement on the right hand side is essentially the movement that the octopus goes through, and it's a complex muscle movement, especially for a hydrostatic skeleton itself. Because there's multiple steps that have to occur. And then this is not even talking about this isn't even including the rest of the tentacles that is needed to maintain equilibrium in the whole entire robot itself. So there's multiple phases that occur and complex steps that occur in this robot that are again are just made of all these different fibers of muscles that are interwoven. So this paper essentially tried to create a robot to try to mimic this hydrostatic skeletal structures to make hydrostatic robots themselves. What they did is they used this electrosensitive material called electrolygra. I'm probably not pronouncing that right, but essentially with its resistance changes based on the pressure and its halves and the interaction with the materials it touches, and that's partly what they use for the tentacles themselves. This octopus model has in the photo there, oh, it's so tiny. Sorry about that. Has 8 servos for each tentacle. It has visual control flow, it has central control flow, and peripheral control, and that's where the CPUs are located. To run this experiment, they put this robot, they called it the Octopus. All caps, so it's probably an acronym that I couldn't find. But they put it into a 260 centimeter by 170 centimeter by 20 centimeter fresh water and had it run a couple of routines. They had it essentially manipulate some objects, run around in a path. They had it manipulate a wooden bar, a round polymetric cylinder, different materials, obviously, different shapes. They basically wanted it to pick it up or just, interact with it and then pick it up, put it down. Around polymetric cylinder and obviously an aluminum metal cylinder itself, and they recorded the times and to see if it could mimic a typical hydrostatic model. What they found in the results is that as you can see that there was a lot of error. However, they did want to point out that it did mimic hydrostatic movement. One of the issues I had, I think, which was pertaining to your question, I think, was this robot itself was not omnidirectional. It can only move forward and backwards and like tilt and turn, which is really weird because in the paper itself, these authors just said that it mimics true hydrostatic movement, but I would answer to what extent and to what it was, because it looks like they just kind of looked at certain measurements and then kind of cherry picked the rest away. And I would think, well, yeah, if I can pick out, it could look like anything. They found that it moves at equal speed independent of a terrain. So they did use multiple terrains for these trials. And they did claim that it's one of the first soft robotics with a push and manipulation, so it can actually pick up materials. They also did note that the time was actually slower than a real octopus going through and running all these parameters. They really tried to sound like this is really it was a huge milestone, but in reality, I think that if we go back to the model. The fact that like one octopus has a giant manipulating arm and can only move in one direction is not really true and the fact that these tentacles are not actually hydrostatic robots kind of just highlights the I guess the ignorance in the field and just how much more knowledge there is to be gained to actually have true hydrostatic robots. This paper was from the University of Jerusalem, I think it was. So they're looking at robotics underwater, which is true, which is interesting. So they're trying to, so essentially, in summary, there's still a lot more to get a true hydrostatic robot. And more octopuses will be needed to run these kind of data and probably get closer to the actual model itself. Quick question I have for everybody else is what biomedical devices have been constructed after animal modeling can you think of besides this octopus? Like
Aqualung?
which one?
Aqualung?
Yeah. That's good. Any others that you can think of, like animal models that have been? There's been robotics, I think, based on crabs, that helps. No, they scuttle. I think that's been a model that people have used. Anybody else? Any other ones?
I don't know, you know those Vibra robots, right, that were, I wonder if they were modeled, I think they were modeled on plants, so.
There've been a lot of insect models, like tiny,
Yeah.
like flying little things.
Wouldn't be surprised if drones based off that. Moving on briefly, I'm gonna go briefly over, while looking at the time, for the neuroscience paper. So this neuroscience paper looked at two different cephalopods. This one on the right is actually a squid brain. That's why it's you can see the matrix is shaped kind of funny. Squid brains themselves are donuts that wrap around the beak inside the so the mouth is in the middle of a squid brain itself. And so, fun fact, squids eat too big of foods. They can actually give themselves an aneurysm, which is interesting. But that's why the matrix is shaped really funny. But what this one kind of looked at, what this review is showing, that there's different areas of the brain that are associated, and this is the central brain I want to point out, not the ganglia itself. That's why I think that this brain gets hurt, the octopus is dead, but the ganglia themselves are fine. But there's different components associated with different behaviors in the central brain itself. So there are functional and structural differences. Another huge difference, I think, with cephalopod brains is their neurons are actually they don't have any myelinated neurons themselves. The giant squid has the largest known axon known to man, and it's just if you can imagine from an electrical engineering standpoint, the Ohm's law must be ridiculous because it's got a huge diameter of a neuron and it doesn't have any insulation around it, which is interesting that they can exhibit behavior and function with that much electrical discharge just going on. But there's also genomics that's been taking place in the field of neuroscience as well. They have identified some genes. I could send more in the chat later. I did this paper somewhere, but they have genes associated with certain regions of the brain differentiation when the cell actually does develop. And then also that's really recent is in 2023, which is just last year. I think it was NIH said that they gave the same amount of rights, or I guess for scientific studies that primates do. So any studies going forward would have to have the same level of the same level of protection, the protection the octopuses have as primates. So there's so a lot of these papers that I went over, they're pretty brutal in the way that they treat the octopuses, and that wouldn't be that wouldn't be applicable with this policy that was introduced in 2023. So especially for neuroscience. So this paper, really briefly, I'm going to zoom through this. So the paper Octopus Visual Systems, a functional MRI model for detecting neurological electrical currents without a blood level dependent compound. Essentially captured a whole bunch of octopuses outside of California. They had 50 grams and 110 gram female. So those are the range of the octopus they found. They This one was 2011, that's why it was kind of brutal with the way they treated the octopuses. But they cut out the occipital lobe and the central brain. They fed a thin copper wire into the optic nerve, into the eye of the brain, and with a sensor attached. And they had a stainless steel wire in the center of the center brain. And they stuck this whole thing into an MRI and wanted to see what activations occurred with the octopus themselves when it saw certain things. So essentially what they're doing is they're trying to see that if the octopus sees something, will there be activation in the brain because sight. They use for imaging parameters, they use a single shock gradient echo. They did produce an EPI for the imaging itself. And here are the imaging parameters they used for their analysis. I'm not gonna say that out loud just because it's a lot and the ton. They did use the regions of the brain. They did run a t-test and they did apply the general linear model to associate the regions that I previously described. What they did find was well, and then what they did, sorry, I forgot about this step. And then they used the Monte Carlo cluster sizes to actually couple the brains together. And they did have a couple findings that they reported. They mentioned some issues with the hemodynamic part of the brain, which I think is kind of a huge issue. They pointed out a whole bunch of artifacts. They claim that these artifacts were due to we're due to the copper, to the copper in the blood, not to the copper that they just implanted in the brain itself. But they pointed out the need for more hemodynamic fluxibility in neuroimaging to study these animals going future. They also did think that I also did want to critique that they also did have through the radio frequency coil, they actually had a really tiny one. And I do think that could affect the results too, because if you're getting like these tiny slices, then it's probably not gonna add up that well. Obviously the voltage gated ions that they did report from the copper itself, they interact with the copper wiring, they said. So the data is probably not the best for usability, but it does highlight the need for more hemodynamic models and imaging itself. They even said specifically that more sensitive superconducting quantum interference devices will be needed going forward for imaging itself. To actually study animals that don't have heme in their heme a subgroup in their blood. So I got a question based on that. Is there any way that you could incorporate octopuses into your research that you guys are doing? Based off that, with any different blood models? Any thoughts? Probably not the best because we don't really.
I mean,
have
the current law would, you'd have to have them in water and alive, right?
Yeah.
To do that.
Yeah, well, so there's a paper that was published in 2023 that I almost included along with the AI one in this paper, but it was a lot. That's why I was like, I probably should cut these. But that one was a model proposed for more octopus to live together for a larger sample size. Because the problem with these last two papers is that their sample size was less than 20. And I think that's an issue. Yeah, sorry.
I'm waiting for you. I'm not trying to interrupt.
Yeah, no.
I was just thinking like hierarchical ensemble model or hierarchical mixture of excerpts, maybe, or some kind. Like their nervous system having like central control, then like still very powerful control, but not central control anymore. I think that would probably be, it might be architectures that would succeed in certain niches for that.
I was thinking about how you can incorporate that as executive functioning, because wouldn't the central control need to like almost override or have input down to the ganglia themselves? Because if each arm is activating with like almost a mind of its own, wouldn't there need to be like a central thing that kind of inhibits the responses in those ganglia? And could that be, how is that any different than executive functioning? So that could be there too. Anyway, sorry, just a random thought I had, but.
Just thinking like the practical stuff of like, so the way you have to, stuff you have to go through for measuring primates, like keeping them in one like fixed location, and that already like affects the dynamics that you're gonna be measuring. And if this is a very different physiological kind of animal, how do you keep it in one location? And it's got arms that are also need to be fixed in one location then,
And they have
that's like a
like,
skeletal structure, so it's like gonna be potentially moving around the body of the organism as well, like for MRI, it's gonna be really tough to measure without doing these like extremely cruel things to these animals, right?
yeah, exactly, because like you need you basically would have to put water inside the MRI, and that has like a lot of water has a lot of electrical interference just because it's a really, interesting molecule. And so going forward with that, it'll be difficult. I don't know if you can get like a tank of water with a certain temperature inside an MRI, or how would that even work? Because you would need to like get around any hydrogen bonds that occur randomly. But
Wouldn't it boil the water with all the magnets?
yeah, probably, actually, it's good. It might, yeah,
It's
well,
like a microwave.
that too. So yeah, I don't know how it's gonna go forward with 2023 that they have primate rights, because I mean, keeping a chimp is kind of cruel too, but not to the extent that it would be keeping an octopus. But there was a paper that maybe I should have included it, that like proposed a model for keeping them, and it just did not seem practical at all, because the amount of money that you would have to pour into to keep the temperature the right way, to keep the stimulus just going, and then to make sure that they're not interacting with each other, because most octopuses cannibalize each other. There's been a new species, yeah, too, right? So it's like keeping spiders. You can't really that's why spiders so hard. You can't really keep them all together, because spiders will cannibalize each other as well. There is a certain group that is recently discovered that's more social, but that was just this year. And so I doubt that anything's gonna come out of that, but my guess would be is that they'd have to use that species, because those are the only kind of octopuses that could keep together. But yeah, it would probably boil.
Why wouldn't they use instead of fMRI, optogenetics method to watch their brains? Because that's not interfere with water.
That's a good idea, man. I think that would be, I haven't looked into that. I can get back to you. I think that's a really good idea though, because yeah, that is my guess would be a level of detail that could be derived from the information. Sorry, cover my mouth. I think that would be part of it.
The standard imaging for terrestrial animals just sounds really tough to apply here. I think we need to rethink how a lot of it is working if you wanna, do MRI or something like MRI on these animals.
Yeah. I'll send you the paper that they proposed for the tank for that. But yeah, it was weird, and I was thinking, yeah, that seems like a lot of issues, but yeah, I should have included it. Oh well. So the final paper, I'll briefly go over. I'm looking at time too. Is briefly the stages of wound healing for regeneration. So in a typical when you typically cut yourself and there's a tiny cut, like say on your arm or whatever, there's usually 3 stages, but I mean we're including 4 because we want to include the actual cut here. So you get a cut, there's usually an inflammation response, which is a negative feedback. Your body senses that there is something changing, doesn't want it to change, and so it signals all these other responses. There's a proliferation phase, which then basically has to essentially just keep out infection and regard cells and anything like that. And then there's obviously remodeling phase, which usually with humans deals with collagen itself, and there's usually there's hypertrophic and atrophic experiences with collagen. That's but I'm not going to go over that for sake of time. The fact that I love collagen so much that I can go over that too much. So so briefly, that is a very very simplified view of how our body heals from scars that are dealt with tissue, specifically in cutis tissue itself. So for that method, they looked into regenerative medicine as well. So octopuses regenerate their cornea, their peripheral nerves, and neural pathways within central nervous system, not the main brain. So just eyes, ganglia, and any pathways that connect the glia and the tentacles, and other nerves. So what this experiment did is they took a whole bunch of lesions and different nerves associated around the octopus's body. Again, another cruel thing to do, but probably wouldn't pass for today. And they watched the regeneration that they had with each one, and they monitored it. So it's an observational study, essentially, with some blocking. I guess they not really observational because they didn't technically count these octopuses. What they found was that in one day there's 12 male and 5 female. Also another issue. There's might be a little bit of gender bias because octopuses do have different anatomical features based on their gender. Their sexual organs are actually located like almost looks like their armpit, really, between the tentacle itself. So there are definitely some gender biases in the study itself. But they found that each cell had when each cut in the nerves itself had 1,800 nerves per stack a day, which is astronomically huge in comparison to regeneration. After 60 seconds of the lesions, these octopuses went back to their normal predatory behavior, so their behavior kind of resumed after 60 seconds of these nerve cuts. You can see in the right hand side, that's the amount of time as well, and the amount of I think it's cells that they noticed from healing, from the healing from the octopus lesions themselves in comparison to each nerve that they cut. And between 1 and 2 days, the octopus its chromatophores actually kind of recovered back to their a similar state. So it was looked brown as opposed to the rest of the body being red. I'll show pictures in a second. And it took 3 to 5 days for all these nerves to heal completely. And if you look at the photos here, sorry, this is a little squeamish for those people looking at these. You can tell it from the cuts. I hope those are big enough. Let's start with the one on the right. You can tell there is a normal octopus. There's an initial cut, and you can tell that based on the nerve, the chromatophores can't really take any signal. So it just looks pale and just really just normal. I guess what a normal octopus would look like if it couldn't control any of the chromatophores. And within was it 1 day, it looked kind of slightly more brown because more chromatophores were healing. And then finally, 5, 6 days of full recovery of these nerves. And you can see the octopus looks pretty much normal. From a staining standpoint, you can kind of see the lesions themselves, and you can actually identify the cells that are coming in and actually healing in the remodeling process, in the proliferation process themselves. You can actually look and see the different type of areola and adipose tissue. If you look at the highlighted F, G, and H sections, and you can kind of tell that there is a lot of cells interacting and healing themselves in the process. And this kind of signals that there is a lot going on, and you can tell the differences between the nerves based on the activity in the region. So there is different healing at different rates at different peripheral nerves, which is interesting too. Okay, and the final question is, this is more of a quiz one. When were octopuses graded the same rates as primates? What year?
Last.
Yeah, thanks. So that's my presentation. Sorry, it's a lot.
Yeah, it's very interesting, and for people aspiring to be xenobiologists, right? First understand what we have here on the planet.
Yeah, because it's complex, and I think as Brad pointed out, like going forward with some of the studies, I think it'll be difficult to study these animals, but the amount that they could teach us is huge, and not just the fields that I described, but more. So yeah. Any questions? Sorry.
And do we have any groups? I know there are a lot of little groups that do primate studies. But I don't know, does MRA do, because they have a primate lab? They don't have anything else like outside of they have mice labs, of course.
So I've been walking to the aquarium every day, every week since I've been here, because it's a 10-minute walk from here. And I did talk to some of the aquarium people, because it's pretty much owned by MRA, they allow interactions. They do say that they would allow Tsunami, the octopus, to be used for studies.
Probably behavioral study there.
Yeah, exactly. I did ask about that though, because I was like, and because I mean, I've been reading to him every week, and it's been and he like clearly identifies me now, so it's kind of fun.
Have you read Remarkably Bright Creatures, the novel?
You know, it's so funny. Somebody mentioned that to me earlier spring, and I probably need to.
It's good. I mean, it's very much what you've described.
Somebody would solve a crime if I was, because I mean, he literally comes out and recognizes me and puts his little tentacle on the glass every time I'm there. So I mean, yeah. So yeah, he would solve a crime. I guess it would be funny, but
It's a good book.
yeah, it's a good book.
Yeah, very cool.
Bet you could study their IQ, like,
Probably.
I mean, the IQ tests for kids are pretty simple.
They have problem solving stuff that they do, because I mean, they're